Okay, g'day everyone. My name is McInerney. I'm a PhD student at the John Curtin School of Medical Research in Canberra at the Australian National University. Today I'm going to speak to you about some of the work I've been doing in using autosomal genealogies to infer evolutionary histories of human populations. So evolutionary histories of human populations were inferred primarily from the 1980s to around 2010 by building genealogies from the mitochondrial genome and the Y chromosome. And these markers were used for their non-recombining nature, which meant that a single genealogy was sufficient to explain the evolutionary relationships of samples at these loci. This revolutionised our understanding of our own origins, and in particular the 1989 mitochondrial genealogy by Carnot was foundational to the out of Africa hypothesis of human origins. However, these are just two loci, and the genealogies of these two loci therefore simply reflect the evolutionary history at these two loci. And as we know, evolution is a random process whereby the same demographic history can result in vastly different genealogies. So to do a thorough and rigid investigation of human evolutionary origins as a whole, we need to have a look at the many independent replicates of the evolutionary process, such as what you find in the autosomes. So why haven't the autosomes been fully utilised in terms of full genealogies? So the autosomes contain a large number of markers that are very useful for evolutionary analysis, but we can't treat the autosomes in the same ways that we could for the mitochondria or the Y chromosomes. And this is because of the process known as meiotic recombination, which means that homologous segments of chromosomal pairs are exchanged. This process disrupts the shared pattern of inheritance between sites. As a result, evolutionary relationships of samples on a single chromosome cannot be explained by a single genealogy. Therefore, we need to find regions or blocks of non-recombinant haplotypes, also known as haplotype blocks or haploblocks. And this is because non-recombinant haplotypes can be explained by a single genealogy because the sites whose shared pattern of inheritance hasn't actually been disrupted by meiotic recombination. But because many genealogies result from recombination, this could actually be very useful for understanding evolutionary history. And it means that we could have hundreds of thousands of independent replicates of the evolutionary process. So in general, there's two ways of inferring or partitioning autosomes into haploblocks that are generally used. The first of which, the four gametes test, also known as the perfect phylogeny principle, is based on the principle that the presence of all four gamete combinations is only possible through recombination. So if all four possible gamete combinations at a site are observed, this is inferred as evidence of recombination occurring between these two sites. This is explained by the figure that you see here. For the ancestral gamete pair, big A, big B, big B mutates to little b along the left lineage, and big A mutates to little a along the right lineage. Therefore, the only way that we can get the little a, little b gamete combination without homoplasy is recombination. The second group of methods utilise linkage disequilibrium. This is based on the principle that sites that have not been disrupted by recombination will exhibit non-random association of alleles. And there's two main ways of calculating LD, which have been D prime and R squared, but I'm not going to go into the details of that. Sites within a haploblock are defined, which is defined by LD, should exhibit greater internal pairwise D prime or R squared compared to sites between haploblocks. And as seen in this figure, LD is signified by warmer colours, and haploblocks can be seen as continuous stretches of warmer colours surrounded by the black box. However, both classes are fraught with some limitations. Firstly, the four gametes test assumes an infinite sites model, meaning that a site can only mutate once, which precludes tri- and quadrillelic sites that we know can be observed in real human data. And all pairwise comparisons that fail the four gametes test are assumed to be separated by recombination, as sites can only mutate once. This precludes recurrent back mutations or homoplasy. So this means that haploblock boundaries will prematurely terminate in the presence of homoplasy. Linkage disequilibrium methods, on the other hand, often have arbitrarily set thresholds of high LD from which haploblocks are formed. This means that depending on the high LD threshold, LD can extend to sites separated by a recombination breakpoint. This leads to artificially large haploblocks that require multiple genealogies to explain the evolutionary relationships of samples. And both of these limitations to otherwise useful and important methods show the need for an approach that can tolerate homoplasy but not tolerate recombination. So we've been identifying haploblocks by utilising a theoretically sound method of testing genealogical congruence, known as character compatibility, which is very similar to the 4 gametes test. However, there's the important distinction that incompatible sites can be explained by recombination, gene conversion, as well as recurrent and back mutations. So it doesn't assume an infinite site model. And character compatibility is actually quite simple. 2 sites in an aligned nucleotide sequence are defined to be compatible if there exists a possible evolutionary history of the sequences in which all nucleotide changes at both sites can be inferred to have occurred only once. Now, as you can see in the figure I presented, each site has a number of possible splits patterns that could explain the evolutionary relationships of the samples. And of course, as I said, character compatibility tests if 2 sites share any possible splits patterns. So if you note, sites X and Y share a possible splits pattern here, and sites Y and Z share a possible splits pattern here. But sites X and Z do not share any possible splits patterns. So for all your sites, you do an all-by-all comparison, and you end up with a compatibility matrix. And it's actually quite simple to interpret. Compatible pairwise comparison in this case is coded as 1, and I've colored it red for just visualization's sake. And an incompatible pairwise comparison is coded as 0. Once you've got your compatibility matrix, we can now go and try to identify haploblocks. And before we proceed any further, this is a compatibility matrix with haploblocks identified. So you should hopefully be able to see these black bars here. And this is made out of real data. Now, normally this plot would be symmetrical, but only the top half is shown so that sites along the haploblock boundaries can be denoted. Sites are, of course, ordered along the matrix according to their genomic positions. Now, in all of the further examples, I've created compatibility matrices in a Han Chinese population from Beijing. And this data comes from the 1,000 Genomes Project Phase 3 dataset, which there's 103 Han Chinese individuals. All the data's phased, so we know which chromosomal pair the allele is sitting on, and I filtered to a minor allele frequency of 10%, to prevent any recent mutations from causing long-range compatibility, which would distort the haploblock finding process. Haploblocks are then identified from the compatibility matrices by applying the CLQD clustering algorithm to them, and this was done by modifying an existing R package BLD and its more recent counterpart GPart. I'd now like to show you how character compatibility compares to LD in haploblock identification. In the figure I've shown here, we have what I think is an excellent example. The character compatibility matrices are presented on the top half of the diagonal and LD on the bottom. The genomic region presented for both compatibility and LD is the same. And you'll note that there's 4 big character compatibility haploblocks here for just the one big LD one. And visually speaking, it's clear that the LD haploblock cannot be explained by a single genealogy, and it's therefore proper to analyse it as 4 separate unlinked genealogies. But what happens if you were to build a genealogy from this LD haploblock? So if you were to build a genealogical network, as I've shown here, you get very noisy structure. If you're not familiar with genealogical or phylogenetic networks, they're very similar to phylogenetic trees, but they display noise in the form of reticulations. Haplotypes can also be displayed as ancestral nodes, and node size corresponds to the number of individuals that share that haplotype. This genealogy exhibits properties expected when a phylogeny is built from a recombinant alignment. That is, the terminal branches are very long, and there's long reticulations across the network, as seen here, as the phylogenetic relationships cannot be properly resolved. Also, there's only a few haplotypes that are shared by multiple samples. Now, given this is a population sample, you'd expect a few common haplotypes shared by many samples. So how does the genealogy inferred from the LD haploblock compare to the character compatibility haploblocks? Now, I've numbered these for clarity's sake, but they just go in order as shown here. And as you can clearly see, the genealogical network inferred from the character compatibility haploblocks exhibits a structure that's a lot cleaner. The terminal branches are a lot shorter, and there's a few common haplotypes that are shared by many samples, as seen here. So we're also more confident in these genealogies because they're identified through a theoretically sound method of genealogical congruence. So now we've seen these genealogies, let's have a look at the evolutionary histories of the Han Chinese population that can be discerned. Now, I've used a really extreme fringe case here, where even the LD haploblock is a little bit dubious. So these haploblocks come from a 460 kilobase stretch along chromosome 18, and the population history was inferred by reconstructing the temporal changes in effective population size using the extended Bayesian skyline approach implemented in BEAST 2. So before I go on, I'd just like to explain these plots to you. The x-axis shows time, with the present on the left working backwards in time towards the right, towards the past on the right. Effective population size on the y-axis is shown on a log scale, and the green ribbon represents the 95% confidence interval, and the jagged line is the median value. The skyline plot is derived. The skyline plot derived from the character compatibility haploblocks is shown on the left, is reconstructed by incorporating all 5 character compatibility haploblocks but treating them as separate unlinked loci, and the one on the right is constructed from the LD haploblock. You note that the population size trajectory is broadly similar, with the population bottleneck ending roughly 30,000 years ago. However, the decline phase is more pronounced in the character compatibility derived plot, in the LD plot with a long drawn out decline. The main point of difference here is the scale. The plot derived from the LD haploblock exhibits an effective population size an order of magnitude greater than from the character compatibility haploblocks. Now, I haven't shown them here, but I performed these skyline plot reconstructions on each of the individual character compatibility haploblocks, and all returned a similar value of 30,000, which is roughly in line with estimates of non-African effective population sizes. So, well, how do these compare to another non-recombining locus, say the mitochondria, as we talked about earlier? So, similar to the previous slide, the skyline plot on the left is derived from the 5 character compatibility blocks from autosomal. The skyline plot on the right comes from mitochondrial DNA in the Han Chinese population. Now, both of them exhibit a population size increase around 30,000 years ago, but due to the smaller effective population size of the mitochondrial DNA, which is typically 1/4 of the autosomes, this marker coalesces recently around the 30,000 year mark, and the autosomes have a much deeper coalescence time owing to their larger effective population sizes. So I'll skip to the end and say thank you all for listening, and especially thank you to the National Centre for Indigenous Genomics for kindly letting me use their grant to perform my calculations on the NCI computer. Thank you.